Every one of the doctors today is going to be talking about HH research. I'm going to kind of frame what I think were some of the bigger themes that we've had over the past few years. This is a field that really has moved very far, very fast. It's a very dynamic field in terms of what's happening. Hopefully I'll frame equally some of the things we don't know, because there is a huge amount that we don't know, and then I've got a few concluding comments about how we might go forward. So if you look at peer-reviewed research publications, this is the track record over the past 50 years or so, and the shape of this curve is really clear. I want to comment, by the way, that all of these research papers, at least in their abstract form, are publicly available. So the website that you see at the top there, PubMed, is an NIH-funded website. There's no password, there's no login, you don't have to identify yourself in either way. It just comes up, just like CNN comes up. And at the very top is a search bar, and you can type into that search bar whatever you want. And if you type in hypothalamic hamartoma, at least if you did a week ago, there were 725 papers, and they're in reverse chronological order. So the most recent is the first one that shows up, and the one from 1955 or whatever is the last one. So for those of you who have the desire or maybe the need to kind of keep up on what's new in the field, every 3 months, every 6 months or so, go, type it in, and then you will see what papers have come up since the last time that you checked in. How common is it? We've got some pretty good evidence now about how frequently it occurs. There is a solid paper out of Sweden where the healthcare system basically captures all patients with any kind of a diagnosis that makes the point that hypothalamic hamartoma with epilepsy occurs in 1 out of every 200,000 children and teenagers. Okay, so this is not the group with precocious puberty only, but HH with epilepsy, 1 in 200,000. And there's a similar number that comes out of Israel as well. And so if you use, World Health Organization figures for 2015, these are the kind of numbers. So basically in the US, there's somewhere around 300 kids that have HH with epilepsy. If we extrapolate the world population and include adults, there's probably around 30,000 patients in the world that have HH with epilepsy. So certainly it is not a major public health issue in the way that hypertension or diabetes would be, but there's a lot of people with this problem and a lot of people who are really challenged by it. We've made some progress in terms of what causes it. So this is the why does it happen? Why does my child have hypothalamic hamartoma? And Angus already mentioned that most patients are sporadic, which is to say there's one child in the family that has it. There are no other physical signs that go with it. And it's not something that runs in the family. But about 5% of all kids that have hypothalamic hamartoma have Pallister-Hall syndrome. So this is a dysmorphology syndrome where there are other features. And as Dr. Wolfong mentioned, that can include extra digits and some GI issues and what have you. And there was a really important clue that came out of that, because those patients are known to have a mutation in GLI3 or GLI3, which is a transcription factor. That's a protein that binds to the DNA and then alters what genes are turned on and turned off. And that's a member of the sonic hedgehog pathway. So keep in mind GLI3 or GLI3, and keep in mind the sonic hedgehog pathway, because I'm going to come back to that in just a second. So a few years ago, there were two groups that asked the question, could there be a somatic mutation in GLI3? So kids with Pallister-Hall have a total body mutation. You can do a blood test, and that's the mutation that they have. A somatic mutation is known a lot from tumor research, that there's a genetic mutation within the tumor, not in the rest of the body, okay? And that's called a somatic mutation. And you discover that by comparing DNA from the tumor to the rest of the body, or to blood, okay? So you look at what the difference is, and that gives you the mutation. So basically, a couple groups address that. The top line is the group from Phoenix. The bottom line, or the second line there, Dr. Wallace from Melbourne. And if you kind of combine those two series, 13% of HH patients had a mutation in GLE3. Okay, so that was kind of interesting. It was proof of principle, meaning that the idea was correct, but we still had 90% of patients or so where that wasn't the answer. So we've kind of moved on a little bit further. This line on the slide and the next slide are not yet published data. So this is going to be presented at the American Epilepsy Society in December. This is a collaboration between Phoenix, Melbourne, and Columbia University in New York. Basically, we're using a more sophisticated technology here. So rather than looking specifically at genes of interest, we're doing a whole exome, or kind of a whole genome, if you would, look. And once again, discovered that at least in a small number of patients, it happens to be 13% of that particular cohort, that there was a mutation in GLE3, which was nice because it kind of confirmed what we had seen previously. But using the whole exome approach lets us look at all the genes, and so we were able to find a few more. And we don't need to really worry about the details about the specific names here. There were a couple of the PRK ACA, and then one each of the two following genes that you see there on the screen. So at least at this point in time, we think there's pretty compelling data that we would understand the mutations for 40% of patients. So that's pretty. That's a good bit of progress forward. Still leaving, of course, probably 60% of the patients where we really wouldn't quite understand. Now, the interesting part about this list of genes is that those are all members of the sonic hedgehog pathway. So basically, the cell membrane is here, and this is a very simplified diagram of a very complicated pathway, which is one of many complicated pathways that the cells have. But basically, a protein which is floating around in the bloodstream here binds to a specific receptor. There's a change on the inside portion of that protein that then creates kind of a Rube Goldberg communication system of one protein changing the next downstream. That's often done by either adding a phosphate group or removing a phosphate group. And then the downstream consequence are these transcription factors here, of which you can see that one is the GLE3 protein binding to the DNA and then changing what genes are produced. And this particular pathway is very important in cell proliferation. So HH is not a tumor or a cancer, certainly, but it is a growth. And so there was abnormal proliferation, abnormal excessive production of cells, at least at one time during early development. So this is emerging as a condition or as a disease, which is a pathway-related disease. Okay, and we're hoping to apply even the next generation of technology to kind of drill down on this a little bit further. So this is an active collaboration that's still looking to see if we can push it a little bit further in terms of understanding why different patients have that problem. Now, as you all know, HH comes in two different flavors. There are those patients that have epilepsy, often with developmental issues or psychiatric or behavioral issues. Okay? There is a tremendous diversity with this condition, tremendous diversity. Okay? So each of you have kids who are unique, and somewhere on that spectrum of how much in the way of challenges the different kids have. About 40%, so some of you in this room, 40% of kids with the form of HH with seizures also have precocious puberty. Okay? The kids that have precocious puberty only usually do not. They don't have the seizures, but they usually also don't have developmental issues or psychiatric issues. So they're usually dealing with precocious puberty as an isolated thing. Okay? So why is that so? Why do we have a hypothalamic hamartoma that causes, really two different, very different sets of symptoms? And as a result of the surgical program, we had the opportunity to look at tissue. Patients are kind enough to allow us, with consent, to use the removed tissue and kind of look at it and examine it. So this particular experiment here looks at the question, is there a difference between the patients, all of these patients have epilepsy, the difference between those who had precocious puberty and those that did not? So that's the question that we're asking. And so the upper line there with precocious puberty and the bottom one without. And for those of you who are kind of sharp, eagle-eyed, you'll notice that it's the same photomicrograph that goes with each, okay? And that's kind of a proxy for me to tell you that we are unable, so far, to find any difference in the tissue between those that have precocious puberty and those that don't, in terms of looking at the way that it looks on the slide, looking at the proteins it expresses, in some cases looking at some of the mRNA, the genetic message that gets expressed. The difference appears to be where the HH is located and where it is attached. So it is location, 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 okay? So we looked at the series, the same series where we were looking at the microscope slides. We also looked at the radiology, and the 100% of the patients that had, remember now that all these kids have epilepsy, 100% that had epilepsy were attached in the immediate vicinity of the pituitary stalk, which is the little thin item coming right down there, okay? And that only 8% that were attached further back in the hypothalamus, not related to the region of the pituitary stalk, only 8% of them had endocrine issues, okay? So there was a statistically significant relationship, the likelihood of having precocious puberty relating to where it attached. So when we look at a scan for a newborn, say, or a very young infant, we can usually make a pretty solid prediction about whether that child will have early puberty, maybe a little bit downstream. We also turned it around and we looked at the issue of epilepsy. And here we basically took 100 patients that have epilepsy with their HH, and we asked the question, where does it attach? And we kind of measured in the anterior to posterior range. And the ground zero, if you will, which is this 0 point in the slide, corresponds to the mammillary body. The mammillary body is that dark structure that's right here, and in this image right here. And the mammillary body is a very small structure that's a very important part of a network that's highly involved in epilepsy. Okay, so and when we looked at these 100 cases, 100% of them attached in the vicinity of the mammillary body, and then to a greater or lesser degree going forward or back. Okay, so. Basically, the working hypothesis is that all H lesions are probably the same. If you're attached posteriorly, you've got epilepsy. If you're attached anteriorly, you've got precocious puberty. And if you happen to have a large lesion, which a lot of kids do, then you basically are at risk to have both. Okay? So it's the location rather than the specifics of the tissue. We wanted to kind of address the issue of why HH lesions cause seizures. So now I'm going to return back to the seizure story. For people in the epilepsy field, for Dr. Wilfong and myself, that's a really interesting question. What is it about that tissue that makes it result in a seizure disorder? We do understand now that the seizures, at least the gelastic seizures, and that's usually what the kids have when they start off, arise from the HH. So here we've got a depth wire. This is the red arrow here, stuck into the HH, and the seizure pattern is arising from the HH and not elsewhere. Okay? It's a dynamic problem because over time seizures can learn to come from the rest of the brain. Okay? But when you start off life with just the gelastic seizures, it's the HH that's the problem. So I want to share with you very quickly kind of the cellular model that we've been able to develop over the past 15 years, I guess now. Taking a look at the tissue, and it's hard to not get a little complicated to detail on this, but kind of just stick with me because we're going to kind of develop a little cartoon that's going to be within this box, which is what I'm using to represent an HH. There's two concepts I want you to hold on to. If from this portion of the talk, if you don't take anything else home, this would be key. You know, what is it that causes seizures? You have to have an imbalance of excitation and inhibition. So something has happened to tilt either that, you know, one way, to tilt in that direction. So either it can be too much excitation of the cells, or you've lost inhibition. Okay, but you've tilted the balance in that wrong direction. But that's not enough. You also have to have a situation where the cells are excessively synchronous. So not only are they firing too much, but they're firing together. Okay, and that's what a seizure is. It's large groups of neurons that are firing too much together. Okay, so these are the kind of cellular principles that we look for when we're trying to explain why a particular tissue causes seizures. This is what HH tissue looks like under the microscope. The anatomy is actually very simple, which is it has clusters of neurons. If you think about galaxies in deep space, you know, not a dissimilar kind of idea. There are regions where they got together to form a cluster. Okay? It varies a lot from patient to patient, but from what we can tell, that's a universal feature of HH. Okay? The neurons are the lacy nuclei with the small dark nucleolus, whereas the glial cells, which are the supporting cells in the brain, are these very dark, densely staining cells here. So we've looked into, you know, we wanted to kind of drill down a little bit on what these cells are all about. One of the techniques that we use is in the freshly resected tissue. So there's a runner that takes the tissue from the operating room to the research laboratory, and they keep that tissue alive for 12 or maybe even 24 hours at times to study it. And part of that is attaching a microelectrode to it. You're going to see some of those recordings in just a second. When the recordings are done, we inject the cells with dye, and then we can visualize the cells. Okay? So HH cells come in two flavors or two types. There is the small HH neuron. The names are not very glamorous because we haven't come up with a better one. But there is the small HH neuron that you see here, and the large HH neuron that you see there. 90% of the nerve cells in HH tissue are the small neurons, okay? And that's these cells that you see here. They're very small cells. They're round. They have very local processes that don't go too far away from where they started. They probably stay largely within their own cluster, okay? And then a piece of information that I'm not showing you here is that they express GABA as their neurotransmitter. So GABA is the inhibitory neurotransmitter of the brain. Okay? And that's an interneuron phenotype for those of you who are familiar with that. Basically, there's a very interesting property about these cells which is not typical for epileptic tissue, which is that these cells have spontaneous pacemaker-like firing. Okay, these cells are firing as their natural condition. Okay, and that's what these microelectrode recordings on the opposite side would indicate. So back to our cartoon for the model. The small green triangles are the small HH neurons. Again, 90% of them are these small neurons with this clustering kind of capacity. They elaborate GABA, and they are firing actively as part of their native state. We've confirmed that in the living brain as well as in the laboratory dish. Okay? So there's a lot of GABA going on there. Remember now that I said that GABA was the inhibitory transmitter, right? So that's very contrary to the idea that we have to have too much excitation. We've got too much inhibition going on here, okay? So let's look at the large HH neuron. These are quite a bit larger. They're usually kind of triangular in their shape. They are more highly branched, and we think that these are projection-type neurons that can send their processes to great distances. Okay, these are excitatory cells, okay, which most projection-type neurons would be. Okay, they turn out to have a very interesting cellular behavior, which is that they have the opposite reaction to GABA. So GABA is an inhibitory neurotransmitter. It usually depresses the activity of the cell and causes it to not fire. The large neurons, these neurons here, have the opposite reaction, which is that they actually fire in response to GABA. Okay? And there's a lot of technical stuff behind why that would occur, and it's actually a very fascinating story, but we're not going to do that part today. So let's come back just real briefly to the model. So remember now that the large neurons are fewer in number. We think that those are excitatory projection-type neurons. There's an unproven hypothesis that it's those neurons that send their processes to the rest of the brain, because of course it has to connect to, get to the outside world. But now all the GABA makes sense, because these large neurons actually depolarize and fire in response to GABA. So it's our kind of working hypothesis that this is sort of the functional unit of why this tissue causes seizures. The issue that we're trying to address right now, and this is actually a paper that's been submitted for publication, is the issue of synchrony. So the idea would be that all of those small neurons have to kind of get together and do their thing. One way that nerve cells do that is through a protein called a gap junction, which electrically links adjacent neurons. They are coupled together like a bridge couples these buildings across the boulevard, okay? And so they do the same thing. And it turns out that gap junctions are present in HH tissue. This is an electron microscope picture here, but this is a gap junction down here below. And then on a pharmacological level, in the tissue slice, if we use a compound that blocks gap junctions, this, again, a little too complicated to go into all the detail, but here we have a recording within an HH tissue slice. We basically have seizure-like discharges which are occurring. We add the compound that blocks gap junctions, they go away. We wash that compound out, and the seizure-like discharges begin to return. Okay? So we think that gap junctions are an important part of the story, leading to that synchrony that I talked about before. So here's that model, kind of in its present form, if you will, where we have a working hypothesis, which allows us to kind of test it in certain ways as we move forward with further research that has both the excitation part and the synchrony part. Okay, so where do, for you guys are at the consumer end of the curve, right? You guys want to know, like, okay, I've got a child with this problem and we really want to help them out. What we hope to see is that the kind of experimentation we can do in the tissue slice would lead to ideas that we can test in patients, okay? And here's a little shopping list of compounds which are proven to impact the activity in the HH tissue slice. Okay. The second column has no names in it because none of those compounds are presently proven to be effective. Some of them we use very commonly all the time, like lamotrigine and levetiracetam or Keppra are names I suspect that many of you recognize. We're not particularly impressed by how they work in real life. Ketone bodies, there's not a lot of information on it. There's some anecdotal information that it may help some kids. And then some of the other things are effectively, not yet looked at. So, but these are ideas going forward in terms of things that we might look at for human trials to try to really be clear about whether that's something that can be helpful or not. I have 2 slides on the treatment side because a lot of our speakers today are going to be addressing, the surgical treatments that are available for patients. And Dr. Wilfong has made the point that surgery is in the future for most kids or most adults that have HH with epilepsy. In 2000, we basically had no treatments. I mean, it was a hopeless situation, meaning there was a lack of hope that something could be done. And in 2015, we have maybe almost too many options about what we might do. Okay, and the point of view that I take is that we want to kind of individually assess each patient for the technique or the approach that's best for them. Okay, but there are no randomized comparative trials to really prove, like at that scientific level, that approach A is superior to approach B. That's usually the kind of thing that we would like to have, and we often expect that for the medications that we prescribe, but surgical trials are, of course, much more difficult to do, particularly for a rare disease. So going forward, it's going to continue to be sort of open label trials, and then, okay, how does that stack up one to the other? Lisa Sobey asked me specifically to talk about this technique here as something that might be on the horizon. So this is the research talk, so this would be like, okay, what kind of ideas might be next? And I do want to make the point again, though, that for you guys, because it's hard to have the perspective on this, the rate at which we have made new treatment discoveries for HH is really phenomenal. Okay, it's been a really sweet spot for new treatments coming online for this condition that didn't have anything 15 years ago. Okay, so this is an idea here, maybe somewhat similar to Gamma Knife. So there's a ultrasound basically delivers or deposits energy in the tissue when it studies it. For most ultrasound studies, it's not anything that you'd ever be aware of, but there's a little tiny increase in temperature. If you use 700 ultrasound transducers all converging at one spot, then that one spot can get a lot of energy and it can really heat up and be damaged or ablated. Okay? Dr. Curry's going to talk about exactly this idea using a stereotactic probe, but this is another thing that's kind of on the horizon and is being looked at. The disadvantage of this technique, and the reason it may or may not have legs as a treatment for HH, is that ultrasound has a disposition to really heat up bone, okay? And of course, HH is right down there at the skull base, and it's very close to things that are embedded within bone that are very important, like the carotid artery and the pituitary gland and cranial nerves and what have you. So this may or may not prove to be something that's applied to HH. Some member of the audience may correct me, but I'm not aware that this has been applied to anybody with HH yet, but it is something that is kind of being thought about by different centers. So just a couple of concluding comments. It is a rare disease and a rare epilepsy syndrome. It's obviously very important to you. And for investigators in the field, for scientists, shall we say, it's an important human model for subcortical epilepsy. Epilepsy usually comes from brain cortex. This is the prototypical condition for subcortical epilepsy. And it also is a model for some things that are really important that you guys struggle with to a greater or lesser degree, which is, why does that seizure problem cause my child to have issues with learning? or behavior, okay? And that does happen with other forms of epilepsy, okay? But HH is sort of a single condition that I think is an opportunity for us to learn a little bit more. So, in other words, the things that we're going to learn from HH are not just for HH, but can apply and give us insights into other forms of epilepsy that are quite a bit more common. Patient care, we've had transformative changes. I think I kind of made that point before. It's a rapidly evolving landscape. I think a macro picture here is that care is not going to be monopolized by one or two centers, but it's going to kind of get back to a dispersed model, which of course is the way it usually is for most conditions. There are, expert centers in every state in the country. But the hope that I would have is that centers that want to do this really do put together the multidisciplinary team, as Baylor has done here in Houston. And I think that the treatment choice, and here I'm talking about surgery specifically, really should be tailored to the anatomy of the patient, which is usually that classification scheme that Dr. Wolfram showed. I think we have some we have some need to kind of think about how we can help disadvantaged populations. The stuff that we're talking about in this room today is really expensive stuff, and it's available to people that have insurance for the most part. As we all know, there's a lot of uninsured Americans, much less people around the world that are also struggling with this problem. Questions going forward. I think we're moving away a little bit from the tissue-based questions. I don't want to say that we're finished with what we can learn from the tissue. We certainly are not. But but we've made some progress. And I think we want to move a little bit towards, I'm going to say, more whole brain type issues. And that's why I'm very excited to hear about some of the imaging talks that we're going to have later today. We don't get to look at the whole brain under the microscope, thank goodness. But we do have tools for doing non-invasive imaging to see what's happening in the brain. And some of those techniques are getting very innovative about what are the networks, what are the downstream issues that we know are occurring in these kids. Is the rest of the brain really normal at birth? We think that it is. I think there's not an absolute answer to that quite yet. Why is it a progressive condition in so many kids? Start off with gelastic seizures, pretty good looking developmental progress. Seizures can get worse, and the kids can take a hit in terms of how they're doing with school and behavior, and can end up with some significant disabilities as adults. A technical issue, how does it really connect? I mean, I've made the point that it's a location-related issue in terms of where it attaches, but exactly what network are we getting into in terms of those attachments? Okay. And then, of course, how can we do better with treatment? So I think this kind of a conference is very valuable and very important. I think scientific conferences around HH are also very valuable. The most recent was in Marseille in 2013. And I would propose that every 3 years or so, that would be an opportunity for the small world of HH researchers to get together. And then the other thing, because it's a rare condition and no one center has huge numbers of patients, we need to get together for collaborative research. This is where the doctors really need to get off the dime and get together to work together, and that would include things like imaging protocols. We can maybe do that MRI scan exactly the same way so that it can be an apples to apples comparison. Patient registry issues, Eileen, I don't know if you're talking about that at some point, but I know you will. I know you will. And then there are rare disease consortiums. HH is a rare disease. HH ought to be at that table, and I know Eileen and the other members of the foundation really work hard on that.